Hello everyone and welcome to this class. In this class we are going to study about bioprospecting. Okay, so I welcome you all to Science 101 PG and please do like, share and subscribe of course for the video. Okay. So let us start by the topic what is bioprospecting. Okay, so here is the definition of bioprospecting, which is that bioprospecting is the process of exploring and utilizing biological resources to discover and develop new products. And those products can be some drugs, some chemicals, other materials, etc. Okay, so if you talk about bioprospecting, okay, so basically, what bioprospecting is that wait, let me take the pencil here. Okay, so bioprospecting is about getting some biological organisms, okay, organisms, and by using those bioorganisms, we extract some important compounds. As I have told you, compounds can be drugs, chemicals, or materials, etc. And uh, basically, those extracted things should be reproducible, and should have industrial applications. Okay, and that is why means after producing in industries, they should come to market. That is the ultimate goal of bioprospecting. Okay, so let us continue and let us first talk about the scopes of bioprospecting. Now these scopes are basically the applications of bioprospecting. So basically it have four applications or scopes, which is first is pharmaceutical, second will be your agricultural, third will be your industrial, and fourth will be your environmental scope. Okay, let us talk about pharmaceutical. As I have told you in the definition, so what we can do is, if we talk about scopes, then in pharmaceutical, we can go ahead use the biological organisms. Okay, these biological organisms in order to discover some new drugs or some therapeutic agents which can ultimately be beneficial to humans so that they can be used, and of course because they are being extracted from bioorganisms, so you can say that these are extracted from a natural resources. If you talk about agriculture, then agriculture is again a very good field, right? So, in agriculture, there is a scope for the development of chemicals such as biopesticides and biofertilizers, which can be used, or you can follow the process of bioprospecting and discover all those things. Now, we will move ahead to the third portion, which is industrial application, right? So, in industrial application, one can discover enzymes, can extract or produce some polymers or some other compounds which again will go to the market. Okay, and from there it will be available for masses to use. Then the fourth portion will be your environmental scope, environmental perspective, and in that perspective we are going ahead with bioremediation and waste treatment. Okay, basically these two terms are almost similar, as there will be some problem in the environment which basically happens due to human activities, for example plastic. Okay, now, if you see the example of plastic, we are trying to discover various microorganisms, some enzyme which can degrade this plastic. Okay, because the plastic is everywhere now and it is a problem. So let us move ahead. Okay, so scopes is done. Then let us go to the what are the sources of your biological resources, or basically from where you can start this whole process. What are the sources that are used in bioprospecting? So we will start first with the terrestrial plants. Okay, now, from the time immemorial, or you can say from ancient times, we are using plants to do various things. We are using the plants as medicines. Like if you talk about the plant Texas, Ech, Echinacea, okay. So these plants have their medicinal value. In India, you use tulsi, mint, okay. They also have medicinal values. So basically, you can use these plants for bio uh, perspective, or you can later further develop anything from that. So basic uh, path followed for this is, first of all, you have to collect the plant, you have to extract the particular compound or an enzyme or whatever you want to extract, and you go for screening to confirm its bioactivity. That is why we can also call it as bioactive compound which can be extracted. Okay, so the second, yeah, after the terrestrial plant we are going to move to the microorganisms. Now, in every biological field, microorganisms play a very important. But in bioprospecting, if you talk about bioprospecting, then bacteria and fungi are the two, you can say, microorganisms which are of immense use to humans. Okay, so if you talk about bacteria, then you have Streptomyces species, and if you talk about uh, fungi, then we have Cordyceps species. Okay, so basically these two are just examples. There are many more which can be used for benefit, human benefit. Now, if you talk about the overall process, then you first have to isolate the microorganism from various environment. Environment can be anything like water, can be land. You can extract the microorganisms from food, from various other environments. Okay, then we basically go for the process of fermentation. If you want to produce some substance or you want to convert the substrate into some products, okay, fermentation will be the process for you, and then you can extract the whatever substance you want to extract. Then third, we will go to the marine organisms. Now if we talk about marine organism, of course we are going to talk of every marine organism. Like you have sponges, okay, ocean sponges. They are organisms. I know they lack many things, but they are useful if you see their commercial value. Okay, and then you have corals, you have marine algae. Basically, after algae, you should consider agar as an example. Okay, then what you do is collect these marine samples or basically organisms. Okay, then you will go for extraction, and again you will check the bioactivity by the analysis. Okay, then we have extremophiles. Now extremophiles are archaea. Okay, so basically they are the organisms that live in extreme conditions. You only have to isolate these organisms, and then you have to go for screening for whatever unique enzyme or other protein you want to extract from them. Okay, and if you talk about the overall process of bioprospecting, then first we will start with the collection. Okay, now in collection, if you are using or you are taking uh, whatever plant or animal or whatever, or that I was talking about ocean or marine life. Okay, so you have to go for field work, and if you are working with microorganism, bacteria or fungi, then you have to go for culturing, or you can say cultivation also. Okay, now, after doing this field work or collecting, or sorry, culturing, the collection process is complete, right? So we will move to the extraction part and screening part. So basically what you do is you extract any substance which are produced by microorganisms. So you can obviously follow various methods or techniques, right? Like you simply, if you want to extract metabolites, like secondary metabolites, then if those metabolites are secreted by the microorganism, then you can directly extract them by culturing the microorganisms. Okay, but you can instead use solvents so that only the desired compound will be mixed with that solvent, and later you can just purify it and extract it. Okay, that is your extraction process. Then after the extraction you should go for testing. Okay, because these compounds are as microorganisms, we cannot identify if what we are extracting is what we want or not. So we will go for testing for if that of that compound. Okay, basically in testing we will check the biological activity by performing various assays. Okay, then if that compound is okay, and we get to know that yes, what we are extracting is our compound, then The last step will be your characterization. Okay, this characterization is like verifying. Okay, so in this you can use two methods. First will be your chemical analysis or chemical characterization, in which you identify or characterize the chemical structure of bioactive compounds via using NMR, XRD or mass spectrophotometry. Okay, and after this you can also go for biological testing if you have bioactive substance. Okay, so in biological testing you test them on animals like mice. Of course that should be done with the permission of your government or concerned authorities, and you should first of course you should test them in vitro, like you can go for cell culturing. Okay, cell culturing, and in that cell culturing, which is in vitro, basically we are performing it in labs. So in cell culturing you should test your compound first, then in vivo, which was within the system, or like as I was saying, like in mice or whatever rat you want to use. Okay, so that will be your biological test. Of course there are challenges while doing this bioperspective process, which the first challenge will be your ethical issues or legal issues. Okay, so in ethical issues you should follow each and every, you can say, rule which is meant for a biologist to be followed, so that there will be no ethical issue. And of course you should go for legal issue, like if you are doing the work which was either patented or have the copyright of some person, then you should take legal permission first so that you won't face any legal issues. And of course don't forget taxes, like if you are opening an industry you are payable, I mean government will take taxes. Then the second challenge will be, if whatever you are your compound you are extracting or bringing them to market, is that compound sustainable or not? Okay, yeah, okay. Degradability should also be considered, so that that compound should be easily degraded if left unchecked. Then third will be your technical challenge, in which first challenge will be your complexity of sample. Like uh, sometimes there are cases when the isolation of the sample is not as easy as you think, or the sample, like some bacterium, like if I talk about extremophiles, then it there might be possibility that extremophiles extraction or isolation can be so difficult that it can be a headache to you. Then the second challenge will be the reproducibility. Okay, so in the point of reproducibility, whatever process you are I mean whatever process you are conducting, you should ensure that the bioactive compounds that you are producing can be consistently produced and tested whenever you are performing the experiment. Okay, then, well, that was it. But before ending, let me give you a clear idea of what bioprospecting is. Okay, so basically, first what you have to do is you have to go for discovering or developing. Okay, discovering of or developing of new products using biological organisms. Biological organisms. What are those biological organisms? As I have told you, plants, then you have microorganisms, extremophiles. Okay, yeah, these are the microorganisms, even marine animals. Marine animals. Okay, then from these, I mean, micro, not just microorganisms, but all these biological organisms, you should find some valuable compounds which can be used in pharma industry, agricultural industry, basically any kind of industry, or should have some environmental value. Okay, everything depends on the value, right? Then, if they have the value, you get to know that yes, whatever I'm going to work, it has some value. Then you go ahead and you start with the process. Okay, process of collection, then extraction, Then after the extraction, you will have its verification. Was the name? It skipped my mind. Okay, extraction. Then you will have screening and characterization, right? Verification. Okay, you will have screening and you will have verification, right? So these are very important aspect for bioperspective, and don't forget that it is a critical approach. Okay, so that's why you can say that this thing is very important if you want to work in biological field and discover anything. So that's it for my class. Thank you very much and see you